Hi, welcome to a quick learning video discussing the science and history behind cloning. Before I begin, I believe I should say this video will not exactly cover evil clones or other sci-fi movie stereotypes you may associate cloning with. My apologies, but I swear this is just as cool. Cloning, as defined by Webster's New World College Dictionary, is the technique of producing a genetically identical duplicate of an organism by replacing the nucleus of an unfertilized ovum with the nucleus of a tissue cell from the organism, or of any various techniques for producing genetically identical DNA fragments, stem cells, etc. You may be wondering how this actually works. In a basic explanation, let's compare sexual reproduction with the asexual reproduction of a clone. In this example, normal sexual reproduction occurs. An ovum from a female we will just call A has 24 haploid cells, while a sperm from a male we will name B also has 27 haploid cells. When the sperm fertilizes the ovum, it leaves 27 haploid cells in the ovum. Both of the haploid cells from A and B join together to form 54 diploid cells. A straightforward math equation. This diagram, however, is representing cloning. The ovum, still called A, has no haploid cells nor any chromosomes. In order for the cloning process to work, the ovum must have all chromosomes removed, so A's genes have no effect on the product. Then we add all the combined diploid cells into any body cell. There is no longer a need for sperm, as all of the chromosomes needed come from another body cell inside what we'll call source B. This results in 54 diploid cells fertilized in an ovum that have no genes from the ovum and is purely a recreation of source B. Now that you know how cloning works during reproductions, let's quickly review how cloning has come to be and how the future of cloning works. Before 1958, no unnatural clones had been made. What unnatural means is no one has reproduced the same organism outside of identical twins, which is a natural example of cloning. In the year 1958, a frog species was successfully reproduced from an embryonic cell. Then, in 1996, two scientists announced the birth of Dolly, a sheep cloned from a mammary cell and a fully grown female through nuclear transfer. The experiment proved an organism can be created from cells other than sperm or egg. Nuclear transfer removes the nucleus of an egg cell and replaces it with a mammary cell. The Dolly was a successful attempt. 277 attempts before her failed, proving cloning is still a very complicated process. What has become of cloning? Despite the advancements in genetic engineering, human clones would become such a controversy. Regulations and laws would have to be put into place before clones could walk among us. Cloning can be used to treat infertile couples and help endangered species someday, but for now, cloning is still a rare and considerably dangerous process. Thank you for watching, and make sure your evil clone doesn't come to destroy you.